Hello everyone, and welcome to today's webinar titled Increasing iPSC Numbers Through Systematic Culture Process Optimization in Bioreactors. This webinar is part of the 6th annual event in the Bioprocessing Virtual Event Series. I'm Stefan and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and is sponsored by Eppendorf. For more information about Eppendorf, go to www.eppendorf.com/bioprocess. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using the live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask the Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen, within the navigation bar, or from the lobby within the event. And now, let's get started. I'm happy to present to you today's speaker, Dr. Benjamin Wolters, research scientist at Eppendorf. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. And now, Benjamin, it's your turn. You may begin with your presentation.
All right. Thank you, Stefan, for this introduction. Hello, everyone. Thank you for attending this webinar. My name is Benjamin, and I'm a research scientist in the applications team of the Eppendorf Bioprocess Center. In today's webinar, I would like to present to you the work of one of our reference customers, who is Dr. Robert Zweigert and his team from the Hannover Medical School in Germany. And their major achievement in that work was the increase of the number of induced pluripotent stem cells through a systematic approach for cell culture process optimization in a bioreactor. So the topic of pluripotent stem cells is quite relevant also for us, but also because more and more companies are focusing their activities towards cell therapy at the moment. And as you all might know, due to their special properties, stem cells have the potential to improve human living conditions. For example, they can be used in regenerative medicine to help healing damaged organs. They can be used for disease modeling outside of the body, or also for drug screening and many other applications. So for quite some time, they have been considered to be a game changer in the medical treatment of patients. However, this comes with a problem, and this problem is the need for large cell numbers. If you want to treat a single patient, estimates suggest that you need about 1 billion cells for the treatment of the patient. And if you want to produce a single product, you will need to produce even more, 10 to the 11 up to 10 to the 14 cells per year, which is quite a large amount, which brings us to the next problem that has to be solved. I'm sorry. And this is the inefficiency of maintaining and expanding cells in a two-dimensional space. The conventional way of growing cells for the longest period of time was to keep them in culture dishes or tea flasks or maybe multi-layer flasks. However, this system comes with multiple problems. First of all, most mammalian cells, just like stem cells, are coming from a 3D environment, like our body, for example. And this 2D incubation, as it has been done, is an inaccurate representation. Because, for example, it enables only the growth of cells into two directions, leading to unnatural cell proliferation, for instance, or non-physiological gene or protein expression. Another problem is control. So usually you have these flask or culture dishes in CO2 incubators, and you can even have them in large room temperature to 37 degrees. The problem is here that in these incubators, you can change the temperature and you can provide usually a fixed CO2 content in the air. But to have a proper development of your cells, you want to have full control of all parameters that influence the cells, right? Because if the parameters are not right or somewhat off, it can happen that the stem cells, do not develop in the direction you want them to. And last but not least, the third problem is a logistical one. So since the surface area of plates and flasks are quite limited, scientists have to scale out the numbers of flasks to achieve these high cell numbers that they require for their processes. And this most of the times leads to scenes like this in the lab, where you have just a large stack of these flasks that you need to handle. And since stem cells are especially demanding in terms of media usage and media exchange, one has to change the media for every single one of these flasks about every day to every other day. Sometimes you even have to wash the flasks to remove debris or dead cells. So it's quite labor intense. And additionally, even if you have that many flasks, that it's not nearly enough to satisfy the demand that would be required for your further processes. So new ways to achieve these high cell numbers need to be developed. And this is where the bioreactor comes in as a solution for the process development of these large cell numbers. A bioreactor offers a controllable 3D growth environment for developing your process and also for increasing cell numbers, which is facilitated by two of its properties. First, its scalability, so you have the ability to develop your process in a small bioreactor, get all the parameters right, and then you can scale it up to a larger vessel with similar geometrical properties, which enables you to enlarge the process and then eventually get higher cell numbers out of it. Second very important feature here is that you are able to precisely control various process parameters that go beyond the possibilities of an incubator. And this is accomplished by a dedicated bioprocess control station, which is attached to the bioreactor. So what are the parameters that we can control and optimize in our bioreactors besides temperature, obviously? And maybe I should mention that when I'm talking about bioreactors, I'm always referring to stir tank bioreactors here. So the first thing that we can control, manipulate, is the pH, so how basic or how acidic your medium is. The second one is that we can add nutrients to the medium. So glucose, for example, is a valuable carbohydrate source which enables cell growth or improves cell growth. Then we can change the gas concentration in your culture by, for example, adding oxygen. And as we're talking about stir tank bioreactors, they come with impellers, which we can control by changing the agitation speed to provide sufficient mixing of the medium or components and of the cells. And furthermore, you can set up your bioreactor in perfusion mode, meaning that you have an outflow of used medium to get rid of detrimental metabolites, and an equivalent inflow of fresh medium, and thereby providing a nice and stable growing condition for your cells. And all these parameters were put to work in the example that I want to show you today by Dr. Zweigert and his team from the Hanover Medical School. And they're working with pluripotent stem cells, and they performed their experiments with our equipment. As you might know, when iPS cells are grown in suspension, they tend to form small aggregates by spontaneous clustering. And these aggregates grow over time, either by continued clustering but also due to cell proliferation. And how did they do their experiments? How were they performing their experiments in the team of Dr. Zweigert? So they used our four-fold stir tank bioreactor system, which we call the DAS box. And this controller operates with up to 4 0.3 liter vessels in parallel. And due to their size, mammalian cells are more prone to shear forces, as you might know, that are imposed upon the cells during stirring. And this is even more the case for these macroscopic cell aggregates that stem cells form. That was why all vessels were equipped with a pitched 8-blade impeller, which was specifically designed with Dr. Zweigert for stem cell applications. And the growth parameters were controlled by the DASBox mini bioreactor system, and the whole bioreactor was set up for perfusion operation mode. But why were they using perfusion mode after all? So this was motivated by a study that was performed before already in 2016, where Dr. Zweigert was also already involved. Here they were testing the growth of pluripotent stem cells in single-use stir tank bioreactors using our BioBlue bioreactors again, in combination with the DASBox mini bioreactor control system. And once they introduced the perfusion mode to the stem cell culture, they were able to achieve cell densities of approximately 3 million cells per milliliter, which is according to the paper, is about 45% more efficient than the batch culture approach they were using before. So Dr. Zweigert and his team went a step further when they used still the original setup with the perfusion culture mode, but they added parameter control. So what they did, they introduced a control pH, dissolved oxygen and glucose feeding, and also agitation control. And they even introduced their data into so-called in silico modeling. And with that, using all these different approaches, they were able to substantially increase the cell density here by more than tenfold, and hence were able to reach close to an astounding 35 million cells per ml in this setting, compared to 3 million in the only perfusion mode runs. So let's have a closer look at how they actually did that. So initially, they started with the uncontrolled conditions, so using perfusion mode only, and a fixed agitation speed of 60 RPM. And then in a step-by-step process, they first added pH control, maintaining the process value at pH 7. Then they added glucose feeding, so additional glucose feeding to the culture, providing more nutrients to the cells. And after that step, they adapted the dissolved oxygen content to about 40% for the entirety of the culture. And just a side note here, we color-coded these different approaches and parameters so that you can find them back in the graphs that we're about to look at in later slides. Okay, let's first have a look at the influence of pH control. So shown in dark gray is the uncontrolled cell condition where pH is not controlled. You can see that it started at above 7, so 7.25-ish, and then it went down already after 1 day, and it never really stabilized, most of the time staying below the optimal pH of 7, which is, so staying below 7 is definitely not ideal for the stem cells. The problem here is that small deviations of the pH can already influence the cell growth and also the properties, as we are about to see in a later graph. The red line represents the pH-controlled condition, also starting at above 7. And then once it dropped below the threshold, the controller jumped in and stabilized the pH throughout the run. And we can see a possible impact of this in the next two graphs. If you look again at the gray uncontrolled setting, we can see that the rate by which glucose is consumed in the uncontrolled setting is slowing down already after 1 day. Once the pH control was introduced, shown by the red line again, we can see that the glucose is consumed faster, which in turn leads to a quicker depletion after 5 days of the culture. And therefore, glucose was added to the culture, which is indicated by the blue line. As you can see, the glucose consumption was comparable to the unfed condition, or during the comparable to the condition where glucose was not added during the first days, but never really reached a fully depleted state. As you might know, the byproduct of the glucose metabolism in the cell is lactate. The more glucose the cell consumes, usually the more lactate is produced. Adding more glucose to the culture also increased the lactate concentration. And if the concentration of lactate is high enough, it has the potential to inhibit the cell growth. So keep this in mind. This will become important in a later slide again. But let's look at the control of the dissolved oxygen. If we take a look at all the other conditions that were not controlled, they all start at about 100% of DO, and the dissolved oxygen content dropped quite quickly to below 25%, which is also not desirable for the cells. Therefore, as shown in orange, the bioprocess control station was used to stabilize the dissolved oxygen content to 40%, almost from the beginning at the culture until the very end. So what was the impact of all these newly introduced controls on the cells? The graph on the left shows a comparison of the 4 different conditions tested with regards to changes in cell density shown by the bars and viability indicated by the lines. During the first 4 days, no significant differences could be observed between the 4 conditions. But if we have a look at the last day of culture, we can see that in the uncontrolled setting, shown in gray, about 3 million cells per milliliter were achieved, which was quite comparable with the earlier study that I talked about before from 2016. And if we look now at the induction of pH control, the red bars, we can see already an increase of about 1 million cells to approximately 4 million cells per milliliter. On top of that, by adding glucose to the culture, we can see an even further increase, reaching 6 million cells per milliliter, already doubling the cell densities compared to the uncontrolled setting. However, if we compare the orange controlled dissolved oxygen condition versus or to the glucose feeding condition in blue, we can see that there's no large difference between the two at day 7. And the authors then looked into possible reasons why they did not detect any further cell growth by fixing this DO level to 40%. And one potential or possible answer they found was when they were looking at the aggregate size. For all conditions, the aggregate size increased over time, but the last condition, so the fixed dissolved oxygen, really stands out here. So apparently, the size of aggregates plays an important role for proper cell growth or proliferation, which, if you think about it, makes total sense. So at a certain size, the supply of nutrients and oxygen just becomes limited for the inner core of the aggregates, causing the cells to cease growing or even start dying. And researchers have found this effect to become especially relevant for aggregate sizes of above 300 micrometer. And this might explain the reduced cell growth that we see here in the dissolved oxygen parameter control. So before continuing with optimizing any process-related parameters, the next step was to reduce the aggregate size to circumvent these problems. So while the outcome of steps 2 and 3 could already be considered to be optimizations, at least in terms of cell numbers compared to the uncontrolled condition, the real optimization steps were performed now, or the first real optimization steps, by modulating the pre-culture conditions of the cells and by comparing different agitation speeds. So as said, they changed the pre-culture conditions by reducing the time interval for cell passaging from 96 to 72 hours before the bioreactor was inoculated. This shorter culture period kept the cells in a so-called subconfluent state at the end of the 2D culture in the flasks, rendering them more proliferative. Then they tested different set points for the impeller speed ranging from 60 RPM to 120 RPM. However, as we know, higher steering speed introduces higher shear forces, and in order to alleviate this effect, a shear protectant substance was added, which is the pluronic F68. Furthermore, the DO control paradigm was changed as well. While it was fixed to 40% before, from the beginning until at the end of the run, now it was only enabled after the DO dropped below the critical 40% threshold. Okay, let's first have a look at what this adaptation did to the cell numbers. Looking at the gray bars, you can already appreciate that even with the standard steering speed of 60 RPM, they were able to push the cell yield on day 7 beyond the 9 million per ml mark, which is yet another improvement compared to the 6 million per ml, which they achieved before. This improvement can mostly be attributed to the pre-culture and triggered DO adaptation. Interestingly, the changes of steering speed did not improve the final yield at day 7 compared to 60 RPM. However, if you take a look at the growth kinetic, so the change of cell number over time, you will realize that 60 RPM, that the 60 RPM condition was lagging behind for almost the entire process time and eventually caught up between day 6 and 7. Now let's take a look at what happened to the aggregate size, since this was basically the major reason for the whole exercise in the first place. So on the right graph, we can see that the aggregate size gradually decreased with increasing steering speed. And they found that 80 RPM was already sufficient to reduce the aggregate size below the critical threshold of 300 micrometer. And taking both results into consideration, 80 RPM was identified to be the ideal steering speed, since it results in high cell numbers with good viability and aggregate sizes below 300 micrometer. So now one could ask, why should I bother then about the aggregate size if my standard steering speed is already sufficient to provide high numbers at the end of my cell expansion? But keep in mind that everything to this point deals with the preparation of sufficient starting material for the actual differentiation run. So eventually, once you have your starting material that you want to introduce into your process, the aggregate size might eventually become a limiting factor again if you don't adhere to this critical threshold of 300 micrometer. So with the aggregate size or with the aggregates at good size and the cell density still going up, they went on to the next step, which was the optimization of feeding and of the perfusion mode. And what they did here was increasing the glucose concentration, because, as said before, glucose is an important carbohydrate source for cell growth. But as also stated before, this in consequence can lead also to higher amounts of lactate. And the more lactate is present in the bioreactor, the higher is the chance that the cell growth can be inhibited. This was counteracted by increasing the perfusion rate of the bioreactor, basically meaning that they increased the flow through of the medium. And this led to more glucose coming in from the beginning, but also the increased lactate level that would be created could be transported out of the bioreactor much faster. As a result, what you can see in the violet, the lactate concentration for the optimized feeding strategy was comparable to the blue line, which is just the non-optimized glucose feeding strategy. So in other words, even though they were feeding a higher amount of glucose into the system, the optimized perfusion mode was able to maintain the lactate levels comparable to the non-optimized feeding approach with less glucose. And again, we see the optimization nicely reflected by the resulting cell density going from 9 million cells per ml that was achieved before to now 18 million cells per milliliter, again doubling the outcome. So these are already tremendous numbers, but the authors decided to go even one step further and included computer modeling here. So to put it simple, or to put it in simple words, what they did, they used the results they obtained throughout all of their previous experiments and fed them into an algorithm, which in turn then analyzed the parameters that were used for process optimization and eventually provided a model with the aim to increase the cell number even further. Basically, they obtained recommendations for parameter adaptation and optimization. This then could be implemented into new experiments in the lab. And these results, in turn, could then be fed again into the algorithm, which then resulted in an even more refined model, and so on. And in principle, you could perform this iterative model building process ad infinitum, but the results we are about to see were derived from only 2 iterations. So including all of their previous efforts and combining them with the in silico modeling, they were able to increase the cell density to an absolutely stunning number of 33 million cells per milliliter, which is, as far as I know, until that point, was an unmatched number for stem cell cultures. And just to prove their point, they repeated the same experiments with 3 different lines and were also able to reproduce their findings in all 3 of them. One very important aspect when it comes to the cultivation of stem cells is the quality control of the cells and to check whether they maintain all of their stem cell properties. To show that the cells derived from their bioprocesses remain stem cells, they analyzed their pluripotency-associated marker expression during all process conditions and found them to be present at high levels during all steps using both flow cytometry and also fluorescent microscopy. Furthermore, they could show that the cells did not acquire any karyotypic abnormalities after cultivating them in the stir tank bioreactor. Just to briefly recap once again, you can see how each optimization step helped Dr. Zweigert and his team to increase the cell numbers up to this very, very high number, which is really demonstrating their expertise in process development. So in conclusion, we believe that this approach can be a great blueprint for the optimization of a bioprocess by the stepwise implementation of parameter control. Yeah, and with that, I thank you for your attention. This is the end of the presentation. We also thank Dr. Zweigert and his team for the permission to share the data. And if you're interested in the original publication, it can be found under this DOI that you can see here. And with that, I'm happy to take questions. Thank you for your attention.
All right. Thank you, Benjamin, for this very, very interesting presentation. Okay, the Q&A session will now start. And if you have any questions, please do so now and click on the Ask the Question box located at the bottom of your screen. And I just browse some of the questions that came in. The first one being, how did you ensure that your aggregates did not settle at such low impeller steering speeds?
Yeah, that's a good question. So in one of the slides, I mentioned that in collaboration with Professor Zweigert and his group, Eppendorf developed a special impeller, this 8-blade pitch blade impeller that is specifically designed for stem cell cultures, which enables efficient mixing while keeping shear force stress low at the same time. So this is really achieved by this newly developed impeller.
All right, I hope this answers the question. Next one is, I heard besides glass bioreactors, also single-use bioreactors made from plastic components are gaining momentum in the industry. Are both suitable for stem cell culture, and what benefits would a single-use bioreactor offer?
Yeah. So in fact, both types are suitable for stem cell culture and both have their benefits. So glass bioreactors are a proven technology out in the field for a very long time with the benefit of the reusability, obviously, as you can autoclave them over and over again and they withstand this process with ease. However, reusing a bioreactor also comes with a necessity of really intense cleaning, which first of all elongates the process and also, if not done correctly, increases the risk of capturing a contamination, right? So a single-use bioreactor, on the other hand, doesn't have to go through this whole process because it comes pre-sterilized. It can be disposed after each run. And yeah, having them right at hand whenever you need them just reduces also the downtime of your systems, right? So this whole preparation stuff doesn't occur. Minimal cleaning efforts, and yeah, also, as said, the decreased contamination risk.
All right. Next question. After achieving such high iPSC densities in such a small volume, what parameters should you look out for when aiming at scaling up the process to a larger bioreactor?
Yeah, interesting question. Well, first of all, it's mostly, or probably mostly, about the vessel geometry and making sure that basically everything that you applied to the small scale can be translated to the larger scale. So vessel geometry plays a role, and the choice of the scalar parameters. So the to make sure that you have the constant power input to volume ratio set correctly, the impeller speed, because this obviously changes as well, since the impellers have different sizes or different diameters depending on the reactor. A constant volumetric mass transfer, such as and also the constant oxygen transfer rate. So gassing your system in the same way as it would have been done with the smaller vessels.
All right. Here we have apparently a question from somebody who's interested in getting into bioreactors. Are there any guides on how to connect and to run bioreactors?
Yeah, in fact, we just launched our new website with a lot of information on the basics of bioprocessing. So here you will find a lot of information that will be useful for you. Additionally, I would like to recommend our white paper, Bioprocessing Basics to you, which will answer a few of your questions already. But if you need more information, or if you don't find what you're looking for, then don't hesitate to contact us.
Yes, thank you very much. Here's one question, probably going back to the pre-culture optimization that you mentioned shortly. What should you look out for when preparing your stem cell pre-culture for the bioreactor?
Yeah, that's a good one. So I think everyone working with stem cells knows about the problems that might come with the preparation of your stem cells. So obviously, you want to make sure that, but I guess that for all bioprocesses, is that your starting material suits your needs, or checks all the boxes in terms of quality, because you know the saying, crap in, crap out. So, for example, confirming the right morphology, the correct marker expression, which is really a prerequisite for stem cells if they're still in their pluripotent state. Yeah, by flow cytometry, obviously. Furthermore, you would need to set the right inoculation cell density. This also can depend on the process that you're aiming for. And, well, most importantly, besides the pluripotency, is probably the viability, right? You don't want to introduce cells that are not happy from the beginning. Then you cannot really expect them to recover if they're already half dead, right?
Okay, looking at the time, I think we have time for one more question, but if your question was not answered here in the live session, they will be answered afterwards. We'll say something later to this. So last question as of now is, in the shown process, a perfusion was applied. How does your software support such complex setups, so the bioprocess software?
Yeah, so basically, this is all supported by the DASWare control. So that is the control software. Here you have multiple options, but the one that would be of most help here would be probably the scripting. So we can support you to come up with dedicated scripts for your purposes. And once you get the idea, it's quite straightforward to do it yourself after some time. But again, our experts can definitely help you with setting this up at the beginning. And yeah, so and also, which is maybe worth mentioning, is in the context for cell therapy, since this is all basically around the topic of cell therapy or preparing starting material for cell therapy, the software is also 21 CFR Part 11 compatible, which is, well, a requirement for GMP process, right? Or for producing material that is introduced for cell therapy research or development.
Okay, that's all the time we have for today. I want to thank you again, Benjamin, for your time and for this nice presentation that you had in petto for us. And before we go, I'd like to thank also you, the audience, for joining today and for your interesting questions. Also, I would like to thank Eppendorf, who sponsored today's webinar. And as said, any questions we did not have time today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand 1 year after today, basically until April 3rd, 2025. LabRoots will alert you via email when it's available for replay. Yeah, the last thing to say is that we encourage you to share that email with your colleagues who may have missed it today. And until we see each other next time, have a good day and goodbye. Thanks again, Benjamin.
Bye, everyone.